Right. Now in the last video we basically discussed about the mechanism of actions of different drugs which act on the bacterial cell wall synthesis process. And especially we discussed that penicillins are and cephalosporins and other beta-lactam antibiotics are inhibiting the transpeptidation step, right? So that transpeptidation step is not done and peptide crosslinks are not made in the bacterial cell wall, and cell wall is not synthesized properly. Is that right? And at the same time we talked about that can in the presence of cephalosporins and penicillins, there is autolysin inhibitors are themselves inhibited and autolysin process become more fast, and that also contribute to the cidal bactericidal action of these drugs, right? Now we will go into detail about the different groups of penicillins and mechanisms of resistance and some important pharmacokinetic facts related with penicillins. Just review a little. In the very first video of penicillin, you remember we talked about the different groups of penicillins. We'll just review that briefly and then we'll proceed forward. So in the penicillins, what were the main group of penicillins? You remember there were penicillins which were the narrow spectrum and there were wider spectrum. We can say penicillins are narrow spectrum and there are just wider spectrum penicillins. And narrow spectrum penicillins were, there were two, mummy penicillins and papa penicillins. There were natural penicillins and they were penicillin, yes, G. Very good, penicillin G and penicillin V, very good, right? And in case of, these were, I think we talked about housewife penicillins, yes. Right? They are moderately smart, they are not mucho smart, but we can say they are right? This is the natural penicillins, right? And then we had penicillins which were really, yeah.
Is the grandmother, yes.
Rather it should be papa or grandmother penicillins,
Yeah,
right? Okay, this should not be up, it should be here. These are very heavy weight, right? Now these penicillins are very heavy weight, they cannot enter into the porins and they are not effective on gram negative at all, and they are not effective in many other bacteria because they cannot pass through the layers of cell wall and they cannot reach to the penicillin binding proteins to do their action. But they can successfully reach to the penicillin binding proteins present on staphylococci. Is that right? And special thing about them were they were resistant to the action of penicillinases. You remember? Or beta-lactamases. We talked about they are having a little bit heavy group. You remember their structure? The R group was little heavy and protecting and beta-lactamase resistant. Right? So this is the papa group, and this papa group is very, very resistant. Right? This is the only group which is resistant. In the family, you can imagine there's papa, there's housewife, mommy, and there are two young, beautiful girls. So only papa is resistant against the penicillinase action, and his basement cannot be damaged. Is that right? So what we can say, what were the groups here? Can you tell me? Very good. Nephicillin, methicillin, cloxacillin, decloxacillin, flucloxacillin, which is less used now, right? So nephicillin, methicillin, nephicillin, cloxacillin, decloxacillin, and oxacillin. So all of them are right strong. And then we talked about the wider spectrum anti-penicillins, and in that group we put the two. There were, yes, the SMART model, right? SMART, which were called extended spectrum. Extended spectrum which was covering more gram negative, and we can imagine them, okay. Right, these are the smart one, and these extended spectrum, right, they could go through the porins of many gram negative. What were the members here? Amoxicillin, and ampicillin. But do you think they are protected? No. So they are susceptible to the action of? Yes, they are susceptible to the action of penicillinases or beta-lactamases. These are also susceptible to the action of beta-lactamases. But this Papa type is not susceptible. It has a big R group which protects it. Okay, it's just to memorize it. Is that right? And now we come to the most smart, right? Which were the most smart? The supermodel. This was the model, and here is supermodel. Right? The supermodel has a big trouble. She is not protected at all. Is that right? She is too smart, and right, and she can pass through even, what is that? Porins of many gram negative, right? Like even can go through, yes, pseudomonal outer membrane. But you had to remember, this one was not able to pass through the pseudomonal outer membrane because it was heavier. That is too smart, right? Due to this reason, these are also called anti-pseudomonal penicillins, like Carbapenem, Piperacillin, Ticarcillin, Aztreonam, Mesalocillin. Now, if you look at this board, there are 3 female groups: natural penicillins, extended spectrum penicillin, and anti-pseudomonal penicillin. Only one is the Papa, anti-staphylococcal. Is that right? So what you have to remember, that whenever you are giving this Papa group, right, or grandmother group or Papa group, this group does not need security of anti-beta-lactamase group. With these drugs, you don't need to give clavulanic acid, you don't need to give sulbactam, you don't need to give what? Yeah. This housewife group, which is basically very susceptible to beta-lactamase activity, actually you can look at, all 3 of them are females. This housewife, mama group, and 2 daughters, one model, another supermodel. All of them are effective, susceptible to the action of beta-lactamases. So, about this mama, she is not bothered much. So we don't provide much security to her, you can understand. But for these 2 girls, we have to have much security, bodyguard molecules. So usually, with this group, we add security of, special security of what? So that there is no undue action to them. What is this molecule? Yeah, they are anti-beta-lactamases, right? You know, there are basically 3 of them are available, which are commonly used: clavulanic acid, sulbactam, and tazobactam. Is that right? And this security, bodyguard, should be with her also. Is that right? So many times when we use, again, very easy to remember, there is a family of penicillins. There's Papa, Mama, and 2 beautiful daughters. Where you need the maximum security? To the daughters, everyone understand it, isn't it? Mama doesn't bother much, and Papa is really Papa. You get it? So, this is about the basic concepts related with the family of penicillins. Now we talk about the resistance against the penicillin, the concept of resistance against the penicillin. When we talk about what are the mechanisms of resistance, as doctors came up with the penicillins, bacteria also gradually evolved, and they brought up different defense mechanisms against our ruthless attacks of penicillin and cephalosporins. What was the most common? About this defense mechanisms in bacteria for the resistance against beta-lactam antibiotics are resistance. There are two types of resistance. There is a natural resistance and there is acquired resistance, natural resistance. For example, some microbes are naturally resistant to the action of penicillins, and some of them are initially susceptible to penicillin and beta-lactam antibiotics, and later on they got resistant. They acquired the resistance. Now, first of all, we'll talk about the natural resistance. There are some microbes which are naturally resistant to the action of penicillins and cephalosporins and other beta-lactam. Can you tell me some microbes which are resistant to penicillin? Yes, ma'am, please. You don't know any. Why don't you tell me all fungi, all virus, all human cells? Do you kill human cells with penicillins? Why? Because none of them have peptidoglycan. So all those microbes which do not have peptidoglycan, or all those cells, either human cells or animal cells or microbe cells, all the cells which do not have peptidoglycan, can beta-lactam antibiotics damage them? So it's such an easy answer, right? So there's natural resistance in, let's suppose, all those microbes which do not have peptidoglycan. No, don't use the word cell wall, because fungi have cell wall, but they are not made of peptidoglycan. Fungi have cell wall which is made of chitin. They are made of chitin, right? Or for example, there is what is this funny bacteria, Mycobacterium tuberculosis. That is a cell wall which has an outer layer of mycolic acid, and mycolic acid does not allow the penicillins to go into deeper. So they're naturally resistant. So natural resistance is seen, you can say, the point which you have to write, the microbes without peptidoglycans, or cells, yes, cells without peptidoglycan molecules, glycan. Now, what are these? Of course, number one, within the bacteria, notable exceptions are mycoplasma. I told you in previous lecture that mycoplasma does not have a cell wall. And if someone does not have a cell wall, can you damage the... Yeah, it is just like that. A woman does not have hair. Can you cut her hair? It's so simple. Is it right? So mycoplasma does not have a cell wall, right? So you cannot damage the mycoplasma by cell wall active agents or penicillin, beta-lactam antibiotics. Then there can be in the bacteria like mycobacteria, lepri or mycobacteria tuberculosis. They are having a very thick layer of outer mycolic acid. This is a very waxy layer. It does not allow the penicillins and cephalosporins to go in, and it does not. There's not much peptidoglycan to be acted upon. Then there are chlamydia. Chlamydia are resistant to the action of penicillins. Why? Because book says it, or there's some reason for that. Because, very good. So Dr. Denise is telling you that basically in the life cycle of chlamydia, a component, some type of it multiplies intracellularly. Now you imagine, if bacteria is present within our cell, can penicillins enter in our cell? No. So can that bacteria be killed? No, it's so simple. Is that right? Then, of course, there are so many examples like fungi. Fungi have cell wall, but that is not made of peptidoglycan. So you cannot damage them by penicillin or cephalosporins. So you understand that there are many microbes which are naturally resistant to the actions of penicillins and cephalosporins and beta-lactam. And of course, you must mention human cells, which is the most important point, that human cells cannot be damaged by penicillin Why human cells are not damaged by penicillins? Because human cells do not have cell wall. They do not have peptidoglycan. And that is the reason why penicillins are so safe for us and so toxic to the susceptible bacteria. Is that right? So in these cases there is natural resistance. Then there are some bacteria which are having peptidoglycan but still they are naturally resistant. The classical example is like Pseudomonas. They are naturally resistant to penicillin G and V. Right? They are many gram negative which are naturally resistant to natural and narrow spectrum penicillins. Why? As I explained previously, that many gram negative bacteria have outer membrane with very small porins, and natural penicillins or this mama papa cannot go through those porins. Is that right? So those bacteria are offering a natural resistance against natural penicillins and anti-streptococcal penicillin. To understand it more clearly, you have to watch the previous videos. Is that right? Then there is acquired resistance. Is that right? There is acquired resistance, and this is what is making a lot of trouble to us. First of all, doctors came to know about that some bacteria started producing enzymes which could break down the beta-lactam ring. And you remember these bacteria are said to be producing beta-lactamases. And if these beta-lactamases are destroying beta-lactam of the penicillin, then we were calling it penicillinases, right? So beta-lactam production, beta-lactam production. Now, let's suppose here is a bacteria, and this bacteria has the capability to produce beta-lactamase enzyme, beta-lactamase. It is not beta-lactam, it is beta-lactamase production. Thank you for correction. Now, bacteria produce beta-lactamase. Beta-lactamase is an enzyme. It means it is a protein. If it is a protein, it should be translated from messenger RNA. It means there should be some DNA. So it means those bacteria which release beta-lactamases, they are having some genetic information to synthesize beta-lactamases. So that information may be present in the chromosome of bacteria. Bacteria have one chromosome or two chromosomes? Bacteria have one chromosome which is usually circular, right? So within this chromosome there may be some, you can say, genetic, some gene within this chromosome. There may be a gene which can make such messenger RNA which can be translated into beta-lactamases. Or in some bacteria, the genetic material coding for the beta-lactamases is not present in the chromosome. Rather, in some bacteria, look here, here is the chromosome, and chromosome of bacteria, and here is extra chromosomal DNA. What is this? Extra chromosomal DNA. Extra chromosomal DNA is called, yes, extra chromosomal DNA is called plasmid. Have you heard of plasmid? Some bacteria have plasmids. Plasmids are extra chromosomal genetic material which can auto-replicate. It can replicate independent of the bacterial chromosome, because all the genes which are essential for survival of bacteria, they are present in its chromosome. And if bacteria receive a plasmid, plasmid may provide new protein molecule to the bacteria and provide new characteristics to the bacteria. So actually beta-lactamase producing gene may be present in chromosome or it may be present in plasmid. Is that right? Now you may be thinking why it is important to know. Let me tell you. Actually, if beta-lactamase producing gene is part of the chromosome, right, of course this bacteria will produce beta-lactamases, and this bacteria and all its progeny cells, derived cells, the cells which are produced by the proliferation of this bacteria will be resistant to action of beta-lactam antibiotics. Is that right? But do you think this bacteria can gift its chromosome to the other cells? For example, if this is E. coli suppose, or Staphylococcus, can it give this chromosome to Klebsiella? No. Now you imagine there is another bacteria which come to your body, or in this bacteria somehow come to your hospital, and this bacteria was having a plasmid, and this plasmid was having a gene to produce beta-lactamases. Now this plasmid mediated, what is this? Plasmid mediated production of beta-lactamases is dangerous. You know why? Because this bacteria, when it is present in patient number 1, this bacteria is multiplying and making lot of plasmids, and then this bacteria may give these plasmids to many other bacteria. So it means that this bacteria has a great secret. There is a great secret of protecting against the beta-lactam antibiotic, which is locked in plasmid, and bacteria keep on proliferating itself or surviving itself, and through conjugation process plasmid may go to the other bacteria. You know what is bacterial conjugation, ma'am? You know something about bacterial sex? You people are not bothered about bacteria. Yeah, bacteria are very sexy. Yeah. You doubt it because you are a human being, so you don't consider bacteria as a partner. But bacteria are very sexy. Look at it. Let me make a bacteria. You don't make fun of it. Bacteria are very sensitive to the negative comments. Look at this bacteria. And there is another bacteria here. This is bacterial chromosome. Here is bacterial chromosome, and here is also bacterial chromosome, right? What happens that, let's suppose this bacteria has a plasmid. What is this? Plasmid. And on this plasmid there is one gene which can code for the beta lactamases, and there is one more gene here. And when this gene expresses, special proteins are made in this bacteria. And when this bacteria makes special proteins, there's something very secret. Those proteins will come to the surface of this bacteria, and a small tube will start growing. Don't get emotional, but it is true. A small tube will grow and it will touch the membrane of other bacteria, and this bacteria will receptively provide a hole. Yes, this is true. And then what happens, right, that this is a gene which gives the order to produce sex pilus. It P for pilus, right? So sex pilus is produced, and then this plasmid open, and then it passes through this and reaches there. And now this bacteria was resistant, and eventually when plasmid have gone to the other bacteria, then other bacteria also becomes resistant to the action of penicillins because it is producing beta-lactamases. So this is an example of plasmid mediated resistance. So resistance may be plasmid mediated, it is dangerous. Why? Because one bacteria can pass the plasmid to many other bacteria, and resistance will spread rapidly. But if resistance is chromosome mediated, can bacteria gift its chromosome to other? No, because if it gives its chromosome, this bacteria will die. Right? So when bacteria are living and having the plasmid, and one plasmid may convert into 100 plasmids or many plasmids, and then bacteria is happy to have what? Conjugation. This sexy activity is called bacterial conjugation. It's a conjugation, right? So by conjugation process, bacteria keep on passing plasmids to other friendly and partners, and then all the group become resistant to beta-lactam antibiotics, because all of them are beta-lactamase positive bacteria, right? So this, but do you think any bacteria will love to give its chromosome to others? No. Right? But sometimes what happens? Bacteria die and its genetic material fall out, and some other bacteria come remove a piece of it and take it away and become resistant. But that is less often activity. Conjugation is far more common, far more efficient to transfer the resistance from one group of bacteria to many other group of bacteria. Am I clear? Right? That is why plasmid mediated resistance mechanism, once they come to your hospital, they will spread very rapidly. But chromosomal mediated will spread slowly. And actually bacteria, beta-lactamase production in some bacteria is chromosome mediated, another it is plasmid mediated. Am I right? No problem into this? Okay. And already you know it, that when beta-lactamases are produced in gram-positive bacteria, they secrete the beta-lactamases in extracellular fluid. And beta-lactamases or penicillinases spread into fluid around the bacteria. But gram-negative bacteria secrete but concentrate the beta-lactamases or penicillinases in their periplasmic space. So antibiotic really reached there, and once antibiotic enter into periplasmic space, it cannot run back. It is caught by a big group of beta-lactamases. Am I clear? Right? So this was something about acquired resistance, that many bacteria, they produce the beta-lactamases and then they start doing the resistance mechanisms. And now we come to another thing, another type of resistance which may be acquired. And what is that? Less often, what really happens, that you have suppose 1 million bacteria and you are exposing them to the beta-lactam antibiotics. You know, bacteria undergo spontaneous mutations. So some out of those millions of bacteria, 1 or 2 bacteria alter the genetic material to produce the porin, and they make the porins tight. If they make their porins tight, they become resistant to penicillins. You are understanding?
Yes.
So this is another way of acquired resistance, that by altering the porins, some bacteria may get or beta-lactam antibiotics. Am I clear? Then another way, of course, if bacteria, if you give with the penicillin clavulanic acid, will that help the problem? Yes. No.
Because the
problem is in the porin. No. But in this case, when you have beta-lactamase positive bacteria, usually what we do, we give either papat group, which is protected against the, which does not, it cannot be attacked by beta-lactamase, or we give these smart girls with the bodyguard. But in this case, if a bacteria is resistant against the penicillins because it has altered its porins and they are very, very narrow, right? Now even you give penicillins with the bodyguard, will that help? No. Is that right? You understand it? Yeah. Okay. Then we come to another mechanism. So what was happening? Bacteria can do resistance either by destroying the antibiotic, beta-lactam ring, or they can become resistant by reducing the entry of the bacteria. Become really naughty. Penicillin keep on entering and they out. They use energy dependent mechanism to kick the penicillin in the ass and out of their near environment. Efflux pump. these efflux pump are present in gram negative bacteria. Negative, right? Out. Concentration to really effectively bacteria efflux pump. But when you use penicillin and cephalosporins too much, more efflux pump, they survive more and others are dead. So all the environmental benefits go to them and resistance spread in the environment. Are you understanding me? And then become the master plan of bacteria. The master plan of bacteria. Master plan is that, actually let me tell you, originally most common mechanism of bacterial resistance is production of beta lactamases. So we were very happy when we made which penicillin? Papa penicillins. They protect their beta lactam ring. So Papa penicillins were killing the bacteria very ruthlessly. Even those staphylococci which were having a lot of penicillinase, then nephecillin, methicillin, cloxacillin, dicloxacillin and oxacillin, they were very effective in those staphylococci which were producing beta lactamases because they were protecting their beta lactam. Is that right? Especially the most important in this group was methicillin, right? And then they were derivative of that. So when doctors covered pharmacist produced the methicillin, right? They were very happy because all those staphylococci which were producing a lot of beta lactamases and killing our patients, and doctors are really in trouble. The doctors were very happy that we were giving methicillin and then its related drugs, and even those staphylococci which were resistant were killed. So it was a wonderful drug. But then, life is life. Eventually you get another bad news. Some bacteria really got nasty. Right? They came with a very clever plan. What did they do? That was the last plan of bacteria, and these days we are suffering with that. What bacteria did, look at this bacteria, right? And in this bacteria, of course, what are these proteins? Penicillin binding proteins. What are these? Transpeptidase enzymes, right? And there are others also. Now, what did we do? That we bring the what is this? This is methicillin or related. And if this bacteria was producing beta-lactam. Do you think beta-lactamases, will this be destroyed? No. Methicillin should be able to kill this bacteria. Should be, isn't it? But we were surprised. A new group of bacteria emerged, and those staphylococci were resistant even to the action of methicillin. So then we started thinking these methicillin resistant bacteria are having some different type of resistance mechanism, because it cannot be resistance due to beta-lactamase production, because methicillin is not sensitive to the action of beta-lactamases. And these methicillin resistant bacteria, especially staphylococci, they started really killing the patients. And doctors were very upset. In any hospital they find staphylococcal infections which cannot be controlled by these, right? They send the bacterial sample to the laboratory and they try the methicillin, and in the presence of methicillin in the petri dish, staphylococci keep on growing, and they got very upset. And they said this very dangerous bacteria, this was called Mr. Sa. Methicillin resistant Staphylococcus aureus. Even to these bacteria, you give these things with the bodyguard, can they work? No. What was the trick bacteria played with us? Actually, the genes, there was a gene which was giving order how to make, what is this? Transpeptidase. This gene got mutant, and this gene made altered, what is this? Altered transpeptidase enzyme. And or we can say these mutations in genetic material of bacteria produced altered penicillin binding protein, or altered what? Transpeptidases. Now beta-lactam ring cannot fit on it. Now you come with penicillin G, or you come with methicillin, or you come with this or that. Can we kill this staphylococcus? No. So we say this is a dangerous staphylococcus, even which cannot be killed by methicillin, and we call this organism methicillin resistant Staphylococcus aureus. What are these? Methicillin resistant Staphylococcus aureus. What is the special mechanism of this resistance? Alteration of penicillin binding protein, right? That is why that on these staphylococci vancomycin is effective, because vancomycin, you remember in previous lecture, vancomycin will not allow the new NAM-NAG PEP unit to bind with the existing glycosylation reaction. Is that right? So when you give vancomycin, right, for example, I'm a staphylococcus which is producing beta-lactamases, as well as I have altered my penicillin binding proteins. Now, can you kill me by penicillin, those cephalosporins? No, because I've altered the target protein to which the antibiotic work, and transpeptidation step cannot be inhibited. But if you are clever, you give me vancomycin. Vancomycin will go and will not allow the newly secreted units of NAM-NAG unit to bind with the existing cell wall components, and cell wall will become weak. That is why methicillin resistant Staphylococcus aureus are sensitive to vancomycin. And then there are bad news. Bacteria came up with the challenge. The worst news which we have about the staphylococci are some staphylococci have altered those enzymes also which were doing transglycosylation, and now vancomycin cannot bind with them, and they are methicillin resistant as well as vancomycin resistant. But our cerebral cortex is more than bacteria. We came up with a different way to kill those bacteria. We give them ciprofloxacin, and these fluoroquinolones and ciprofloxacin, they go into bacteria and digest away all the, I will tell you how in later lectures, how fluoroquinolones enter into bacteria, and eventually bacterial DNA is fragmented. This is not the antibiotic which fragment the DNA. In the presence of fluoroquinolones, bacterial enzymes do fragmentation. How they do, we'll talk it later, right? But one thing which you have to come to know, that This is the super bug with super resistant, and this is called by another resistance mechanism by alteration of penicillin binding protein. This mechanism was originally discovered in methicillin resistant Staphylococcus aureus. Now this is also discovered to be present in Streptococcus pneumoniae. You know, the most effective drug for Streptococcus pneumoniae was Penicillin G. Of course, every doctor knows, isn't it? Penicillin G is a killer to the Streptococcus pneumoniae, because doctors were very happy. Why? Because Streptococcus pneumoniae were not producing beta-lactamases. So we could give the Penicillin G and kill them. Is that right? Now the new strains of resistant strain of pneumococci are there, Streptococcus pneumoniae, which are resistant to Penicillin G. Is that right? Why? Because pneumococci has altered their penicillin binding proteins. So even though alteration in penicillin binding proteins was originally discovered in methicillin resistant Staphylococcus, but now we know there are many other bacteria which are using the same strategy, so that our beta-lactam antibiotics fail. The very dangerous changes in the Streptococcus pneumoniae, which is a very common cause of pneumonia, right? And previously we could give injections of penicillin G and very effective response. Now sometimes we are in trouble. Then another bacteria has done also the same thing, and that bacteria may be resistant to, they have altered their penicillin binding proteins and other proteins to which the vancomycin could even bind. What is that bacteria? Have you heard of Enterococci? Enterococci. Many Enterococci are altering their penicillin binding proteins. They are also getting resistant to beta-lactam antibiotics. Is that right? So what are the four mechanisms? Number one, destroy the beta-lactam antibiotic before it reaches bacteria. Before it enters into bacteria, you destroy it by beta-lactamases. Number two, tighten the porin, don't allow it to enter. Number three, if it enters, then you throw it out. It's really a very disrespectful way that antibiotic come happily and you throw it out and again come in and throw it out. Is that right? So, and the last but not the least, very important, okay, let the antibiotic come. Don't destroy it. Let it enter. Don't throw it out. But it cannot bite on penicillin binding proteins because they have become altered. Is that right? So this was some discussion about the resistant mechanism which are present in against the penicillins. Now let's talk about few important points related with pharmacokinetics of penicillins. I will not go into detail, but there are some important points we must discuss about the penicillins. All of you know that some penicillins are given orally and other penicillins can be given parenterally. Is that right? Some penicillin can be given orally, and the others which can be given parenterally, intravenously or intramuscularly, and there are still other penicillins which can be given orally as well as parenterally. Parenterally mean not through GIT, right? Now, the penicillins which are given, which are available oral, which can be given orally as well as parenterally, which are given only orally. Write it down. Penicillins which can be given only orally. Right? Can you tell me the penicillins you must be knowing which are given? Yeah? Ampicillin is available in injection form, ma'am? No, it is. Yeah. Okay. Ask any bacteria. Yes? I think penicillin V. Right? Why don't you tell me penicillin V? Penicillin V, that is given orally because it is acid stable, but it is used very less commonly, usually for the mixed infection in oral cavity, right? Or penicillin V is also used in children with a history of rheumatic fever to protect the further attacks of Streptococcus, Beta hemolyticus, Lancefield group A, rheumatogenic strain. Is that right? Even though these days we prefer to give them intramuscular injections of benzathine penicillin. Right, we'll talk about that later. So oral, some penicillins which are available only to be given orally, that is penicillin V as well as amoxicillin amoxicillin and amoxicillin with clavulanic acid, amoxicillin plus clavulanic acid like augmentin, and carbenicillin in special form which is called indenyl carbenicillin. You remember anti-pseudomonal? These are which are specially given orally, right? Then there are some which are specially given parenterally, right? So A group is which is specially given orally. I'll tell you B group of penicillins which are specially given parenterally or intravenously, Now in this group there is ticarcillin, carbenicillin, piperacillin. So it means straight away these three. Which one? Anti-pseudomonal. You remember carbenicillin? Carbenicillin, you know James Bond action. James Bond come on a car, it has a pipe and throw the tick sometime. So carbenicillin, piperacillin, and ticarcillin. So these are anti-pseudomonal and penicillins which are which cannot be given orally. There's only one out of this which can be given orally is carbenicillin with indenyl group. Is that right? All of them are given parenterally. Or if with these drugs we have added the tazobactam or sulbactam or clavulanic acid, again they are given parenterally. Is that right? They can be given parenterally. So ticarcillin, carbenicillin, piperacillin, or of course ampicillin with sulbactam. Ampicillin plus sulbactam. You know, ampicillin was extended spectrum here, so it need to be protected, right? Carbenicillin, then piperacillin and ticarcillin, they have also protective molecule which are anti-beta-lactamases. So ampicillin with sulbactam is only available in parenteral use intravenously or intramuscularly. Is that right? Now, what about the remaining penicillin? Remaining can be given, are available both oral as well as parenteral. Is that right? Okay, one more point which I will tell, that when you are giving penicillin orally, I told you previously, penicillin B is acid stable because its R group protect the beta-lactam ring from the attack of acid. Penicillin G is not much acid stable. I told you that stomach acid will destroy the penicillin G because penicillin G is sensitive to the action of the acid. Is that right? Then we come to the penicillins which are available in depot form. Penicillins which are available in depot form. Which can be given one injection which keep on working for a long time. There are some penicillins which are injected into intramuscular, deep intramuscular injection, and from there active penicillin is released very gradually, and it's very long acting penicillins. Which are these? Pardon? The procaine penicillin, and what is other? Benzathine penicillin. So, if you add benzathine with penicillin or procaine with penicillin, then penicillin stick to the procaine or stick to the benzathine. And in this combination form, penicillin is released from benzathine or from procaine molecule very gradually. So it keep on working. For example, benzathine penicillin can keep on releasing the penicillin for up to 3-4 weeks. That is why in patients with rheumatic fever, children who have a susceptibility to rheumatic fever, every month we give one deep intragluteal injection of benzathine penicillin. Am I clear? Okay, I just want to know, this penicillin which is sticking to procaine or to the benzathine, which is which penicillin? Mama penicillin, Papa penicillin, or model penicillin or supermodel penicillin? No, look, it is being released gradually. Which penicillin is complex with procaine or benzathine to get the depot form out of these 4 groups? It is penicillin G. So yeah, you are very right. That penicillin G is complex with procaine or normally penicillin G has a very short half-life. But penicillin G, when it is complex with procaine or with benzathine and then given deep intramuscularly, it keep on releasing from there for a very long time. Is that right? So these are two depot penicillins. About the absorption of penicillins from GIT, right? Especially I'm interested to discuss this issue, amoxicillin and ampicillin. Amoxicillin and ampicillin, which is better absorbed from GIT? Very good. So it is from GIT, right? And here you have amoxicillin, MO, and here you have penicillin. So amoxicillin can be absorbed, complete absorption from the GIT to the blood. Penicillin from GIT absorption is very poor. Most of it moves forward. Is that right? It is good or bad? Look, first let me put the question. Amoxicillin absorb mostly from the GIT, right? It is good or bad? It is good and bad. Let me tell you why it is good and why it is bad. If you are treating some infections in which antibiotics should absorb from GIT and go to the blood, then amoxicillin is wonderful. For example, if patient has amoxicillin susceptible bacteria in the throat or in the ear or in some other part of the body, he can take the amoxicillin orally and it will be very good absorption and it will go to the systemic circulation and find where is the culprit bacteria and kill it. But it is bad also in one way. You know how? If you are having bacteria in the GIT, for example, you have Salmonella or Shigella and playing havoc in your GIT, you give them amoxicillin and it does not stay in GIT and gets absorbed. Can it eliminate Salmonella or Shigella? No. So it is good and bad. It is good when you are treating those. It is good that it is absorbed because for systemic, when we require the amoxicillin systemically, but amoxicillin fail on the GIT tract infections. Am I clear? Opposite to that, when we talk about ampicillin, this was what? Amoxicillin, ampicillin. Ampicillin is poorly absorbed from GIT. So when we really want to give ampicillin for systemic infection, we prefer the injections. But we prefer injections. But if someone has a heavy infection in the GIT like Salmonella, Shigella, and which is sensitive to ampicillin, we can give safely oral ampicillin, and ampicillin will keep on going down and down in GIT with poor absorption and stay in GIT and damage the bacteria. So you understand? So both of these antibiotics, amoxicillin and ampicillin, have advantages and disadvantages of this has a good absorption from GIT, this is poor absorption from GIT. Any question up to this? Another point which I want to mention, these papa antibiotics. What are these papa antibiotics? Methicillin, which is not used these days, but nefcillin, clozacillin, diclozacillin, oxacillin, these are anti-staphylococcal antibiotics. Many of them are available in oral form, but you have to be very careful that their absorption is affected by the presence of food. Their absorption is affected by the presence of food. They should be given when patient is empty stomach, either 1 hour before food or 2 hours after food. 1 hour prendially or 2 hour post, right? So they should be preferably given, papa. Food should not be there. It needs its own independent activity to be absorbed. Is that right? Then once these antibiotics are absorbed, of course, they will go to circulation. And in the circulation, how they get distributed in the body. Suppose this is your general circulation. Here was your GIT absorption, which we have been discussing. Is that right? And then once these penicillins are present in circulation, how they are distributed. Remember, penicillin molecules are not so lipid soluble to pass through our cell membranes. Due to this reason, intracellular bacteria are not attacked by penicillins. Penicillins are very well distributed in extracellular fluid. They are distributed in the blood, as well as from the blood they leak out into interstitial fluid. They also leak out into many other body fluid. About the meningitis or encephalitis, when you require the penicillin, there is a blood-brain barrier. Normally, penicillins don't cross blood-brain barrier. But when there is meningitis or encephalitis, right? Blood-brain barrier is broken because in inflammation, what happens? Chemical mediators of inflammation are produced and endothelial cells of cerebral capillaries shrink. Blood-brain barrier is produced by, you know what is the mechanism of blood-brain barrier, or you don't know it? You know it or not? Hurry up. You want to know or you don't want to know? Okay, you want to know. Let me tell you what is blood-brain barrier. Look, here is your central nervous system, right? Here are your neurons, right? And here is, suppose your blood vessel passing, this is the arterial side and this is the venous side. And let's suppose here are the capillaries. And these are which cells? Endothelial cells. This is cerebral capillaries. Now from where the blood-brain barrier concept came? Actually, you know, there was a doctor who was injecting the cats, C-A-T-S, cats with a special color called I1 blue. And after that, he was very ruthless, I think sadistic person. He will kill the cat and look in which part of the cat the dye I1 blue has gone. He was surprised that I1 blue, which injected in the blood of the cat, it will go to the all part of the cat, but will not enter into the brain. Brain of the cat will not turn blue. He said there is some blood and brain barrier. That was the original concept. There is some barrier between the blood and brain, and every molecule from the blood cannot enter into brain. Later on we found that this barrier does not allow many drugs. For example, this is a drug, penicillins. When they pass through the cerebral capillaries, they cannot pass through this and come into interstitial fluid of the brain. And they enter from arterial side, pass through the capillaries and go out through venous side. And they do not enter into substance of the brain. So we say between the blood and the brain fluid, there is barrier. This barrier is actually made by the very tight interdigitation of the endothelial cells. Brain, capillaries, endothelial cells are very tightly linked with each other. Let me tell you, if this is one endothelial cell, this is another endothelial cell. Actually, in the central nervous system, they are interdigitating very tightly, so drug cannot pass through this. But when there is inflammation, and of course in inflammation there are many chemical mediators of inflammation, and those chemical mediators of inflammation act on endothelial cell and endothelial cell shrink. When endothelial cells shrink, what happens? It opens the way for the larger molecule. So penicillins normally cannot cross the blood-brain barrier effectively, but when there is infection in the brain, blood-brain barrier becomes leaky. We call it becomes leaky, and then penicillins start entering into to the central nervous system. And as infection terminates, as inflammation goes down, concentration of chemical mediators become less, blood-brain barrier re-establishes, and then again penicillin cannot cross. So anyway, we are happy that penicillin can enter into central nervous system when they are needed there. Is that right? Another thing, penicillins can, same is true about the bone infections, that in the bones also in deeper part of the bones, penicillins normally cannot enter. But when there is osteomyelitis or more infections in the bone, penicillins can enter. Penicillin can cross the placenta very effectively, right? And because they can cross the placenta, they go to the product of conception. And the good news is that they are not toxic to the developing embryo or product of conception. So they are not having any teratogenic action. Is that right? Then about the penicillin, one more thing is about their metabolism. The very good news is they are not metabolized much. So we don't need to remember how they are metabolized. Yes, once they enter into your body, eventually they go out of your body. It means they are excreted or eliminated from the body. Most of the time they go through kidney. Most of the penicillin go out through urine. Nephicillin, and with the nephicillin there is mainly it's the nephicillin which goes through the liver, right? But most of the penicillins go out through kidney. In the kidney, of course, there are nephrons. What are there? Proximal convoluted tubule, and this is the nephron, right? And here's the drop of urine. Now, penicillin freely filter, but only once they are filtered, they cannot be reabsorbed. So 10% of the penicillin go out into urine through the filtration process, right? But really, in this proximal convoluted tubular cells here, what are these cells? Proximal convoluted tubular cells. They are having very special pumps here, and these pumps are basically, these pumps are called organic acid secretory pump. What these pumps are doing, blood passes through this peritubular capillaries. When many organic acid, when they are passing through this area, organic acid, when they are passing through peritubular capillary system and into this area, this pump will take and actively transport peritubular, from peritubular fluid, these pump in the basolateral membrane of proximal convoluted tubule actively transport the organic acids to the lumen of nephron. The same mechanism is also used by penicillins, and 90% of the penicillins actually bind with these pumps and go into nephron and eventually appear into urine. Now you may be thinking, why it is so important to know this thing? There's a very important fact. Let me make it a larger diagram so you understand. I just make one cell, one basolateral cell, and this cell is having here a very special type of pump. And what is this pump? This is active transporter, which will take the organic acid and eventually throw them into, transport them. Clear? Actually, like benzyl penicillin, or penicillin G, it has a very short half-life. And if we want prolonged action, we give another drug. And along with the penicillin G or with other penicillins, you can give another drug, and that drug should come and start using this mechanism for excretion. The blue drug along with the penicillin goes, and that occupies these transport mechanism. Do you think now penicillin can go really out? No. So this penicillin secretory mechanism is used by the blue drug, and now penicillins will be going back to the body. So they will not be eliminated so fast, and that will prolong the half-life of penicillins. Right? You tell me the name of the blue drug. No, procaine. Procaine was different, which was coming as in injection with penicillin, so that procaine will bind with the penicillin and release it slowly. That is probenecid. Have you heard of it? Probenecid. This blue drug is probenecid. So along with the penicillin, if you go give probenecid bind with the organic acid transport mechanism more strongly than penicillin and occupies those transporting mechanisms, keeps them busy. So penicillin doesn't have a chance to use that pathway to slip out of the body, and they stay in the body for a longer time. Is that right? And of course, like many things, penicillins also appear in the milk. Of course, only in the female usually, right? Do you think it has any implication? Or just I said out of my personal interest? Yeah? Yeah, for breastfeeding. What is the importance of this concept? If mother is taking heavy doses of penicillin for her own infection, sometimes, not always, penicillin may be taken up through the milk by the baby, especially ampicillin, and baby GIT get loaded with the ampicillin, and most of the bacteria in the baby GIT is destroyed, and super infections may develop there, and baby may develop severe diarrhea. But anyway, this is not so often. It is not having much clinical importance, but penicillins are also secreted in saliva. So if you really love someone and you are taking penicillin, you can provide the person with some oral protection against the bacteria. Is that right? So here we stop today. Next time we will talk about adverse reactions related with the penicillins, and then cephalosporins and other beta-lactam antibiotics. Is there any question? Class dismissed.